Good morning, good afternoon, good evening, depending on where you may be joining us at this great event today. Welcome to all summit attendees. My name is Tim Evans. I'm the director of the McGill School of Population and Global Health, based in Montreal, Canada. And it's a great pleasure for me to host and moderate this session of the Healthy Longevity Global Innovator Summit Award this morning, Eastern Standard Time. I would like to say that I'm here representing my better half, my co-chair, Dr. Roderick Pettigrew, who's the Chief Executive Officer of Engineering Health and the Executive Dean for Engineering Medicine at Texas A&M. He and I had the privilege of serving on the Health Longevity Catalyst Award Committee as co-chairs. And I can say on his behalf with full confidence that it has been an incredible privilege to learn about the diversity and robustness of innovation that is bursting at the seams around the world when we think about the challenges of healthy aging. So today we have a number of awardees of the Catalyst Award, and Catalyst awardees are given a modest seed funding of $50,000 for an early stage project that's particularly innovative and aims to extend the human health span. And we have a number of those awardees who are going to give us a taste of what their creative research energies are being directed towards. So we'll listen to their reflections relatively briefly. And then following those presentations, we'll enter into a moderated discussion. And that's the opportunity for all of those who are attending the summit to raise questions. And those could be questions that are directly related to the presentations or indirectly. If you feel that there are tangents that you would like to make that reflect your own research, or perhaps identify new opportunities for moving things forward, those reflections and comments are welcome in addition to specific questions to our panelists. And I'm sure our panelists will have thoughts beyond their research, which will make it an interesting discussion. So with that, let me just describe the outline of our session. We'll be beginning momentarily with presentations from our panelists. We do have one panelist who may be lost somewhere in the ether. And so we're hoping that panelist will be able to join us. And if and when that panelist does, there'll be appropriate introductions. But just to say that we may be having an additional individual join us, an individual awardee join us in the course of the panel. But the agenda will then be for us to have brief presentations from our panelists, probably 7 to 8 or 9 minutes each, so that you get an idea of who they are and what they're focused on, and we trust that after they've presented, we'll be able to enjoy an active discussion with respect to the work that they have generously shared with us. So with that, what I'd like to do now is briefly, very briefly, introduce our panelists, because they will give you more background with respect to who they are, where they're coming from, and what they're working on. But we have two panelists today, Jakkrapatr Baiyokvichit, who's joining us from Thailand, who will be reporting on his project Lab on a Chip for Cardiovascular Disease. And Jakkrapatr will be followed by Ryo Sugimura, who's going to talk about his project on reverse inflammaging by target nuclear PD-L1 in inflammatory macrophages. With that, I'm going to hand it over to our first speaker, Jakkrapatr. Welcome, and thank you very much for making the time, and congratulations on your Catalyst Award. Over to you.
Okay, hello everyone. First of all, thank you for giving me an opportunity to be here. And thank you, Dr. Tim Evans, for the introduction. And my name is Jakkrapatr or Baiyokvichit. And yeah, I come from Thailand. And I'm currently a PhD candidate from National Taipei University of Technology in Taiwan. And my project here is lab ownership for cardiovascular disease, yeah, as Dr. Tim mentioned. And I would like to share in today about how my research could help democratize the healthcare solutions and contribute to achieving the healthy longevity and equity. So first of all, let's start by addressing the fact that cardiovascular disease is the leading cause of mortality globally. According to World Health Organization, cardiovascular disease that take more than 17 million lives each year. This current challenge we face is that typically the traditional method for diagnosing cardiovascular disease require laboratory-based tests like a blood test, and which can be both time-consuming and expensive. And this often limit the access to those in remote or underfunded areas. And moreover, the burden on healthcare system often result in overcrowded hospitals, leading us to the long waiting time for the patients, all right, to see the doctor. And as a result, that individuals may choose to ignore the symptoms like a chest pain and continuing with their daily life, unaware that it could be their last day. Unfortunately, this has personally affected to me as I lost 3 of my relatives to cardiovascular disease. And this personal experience is the driving force behind my commitment to this project. And this is where the lab-on-chip technology comes into play, and my research focus on developing this lab-on-chip, as you can see, and that utilize the aptamer conjugated magnetic nanoparticle swarm to detect the key biomarker for cardiovascular disease, including the cardiac troponin I and brain natriuretic peptide and creatine kinase MB, 3 of this. And together with the advancement of micro electromechanical system that we have in Taiwan, we are moving toward to minimizing the complex laboratory instruments into this compact and portable device. And basically, we just use this one together with the Bluetooth module, and it's an electrochemical device like a potential stat that can transfer the signal or the result into your mobile phone. And by using this device, it will allow individuals to test for cardiac troponin or cardiovascular disease biomarkers at the point of care settings. providing the immediate and accessible health diagnostic. And beyond that, through the careful optimization of the manufacturing process, as we know that this kind of technology is widely available, but it's pretty hard for us for everyone to try to commercialize it because the cost is too expensive. I mean, the production cost or manufacturing cost is unable to commercialize this kind of device and make it accessible to everyone. So in this project, we already we have successfully reduced the cost for each of lab on a chip device, as you can see here, to the approximately $3 US, and this significant reduction in cost can pave us the way for large-scale production and distribution, and it's making our technology accessible to communities around the world. So to conclude, my work on this lab on a chip for cardiovascular disease is more than just technological advancement, but it is a step toward the democratizing healthcare by making cardiovascular diagnostic faster, cheaper, and more accessible. And we can create the future where no one is left behind in the fight against this cardiovascular disease. Together, we can achieve the health longevity equity and ensuring that everyone, everywhere has the opportunity to live a long and healthy life.
Great.
Mhm.
Super. Thank you, Jakkrapatr. And I'm sure the relatives that you've lost would feel like you're honoring their legacy in the most profound way. Terrific work. One very brief question. Can you just clarify what the biosample is that's used?
On the biosample, yes, of course. Because 3 of the biomarker that we use for this lab on a chip is contained in blood. I mean, the concentration of this biomarker in blood for the patient who have the cardiovascular disease will be different from the healthy people. So that is why we mainly use blood currently. We use blood to just to prick and drop it on the lab on a chip, which means this process is could be kind of minimized and kind of easy, similar to the rapid test kit during the COVID-19, and that is why the user can test the blood by using this kind of device without the specialist or without a specialized knowledge in this field.
Sure, great. And just if you could say a comment or two. So a finger prick blood sample is the biosample, and that gets put on the chip, and then you get a readout on those biomarkers.
Yes.
Can you speak to the sensitivity and specificity of those of that mechanism? Meaning, what degree of false negatives might you have if in this, or false positives? Have you assessed that?
Because for currently, we already achieved the limit of detection, and all of the limit of detection is lower than the cutoff value for the guideline. And let me tell you in detail. Like we have 3 biomarker, all right. For cardiac troponin I, the limit of detection is only like 0.032 picogram per milliliter, and for BNP, it's just 0.041 picogram per milliliter, and the final one is CKMB is 0.4 0.05 picogram per milliliter. Like currently, we already achieved the pretty low limit of detection, the acceptable limit of detection. But for the sensitivity is still pretty high. But of course, we use this one as a screen test, which means it's not we still do not have to obtain the very high specificity, but that is the main purpose of lab on a chip, that is make it accessible to everyone and affordable. And but I believe that in the future, of course, we still keep trying to develop our lab on a chip here to increase the specificity and sensitivity. Yeah.
Great, great, excellent point. And I just want to reinforce it for the summit in a widespread diagnostic tool that is used primarily for screening. The sensitivity is so important. You want to minimize false negatives, and if you do that, then if for some reason there's a false positive, further testing will pick that up. But this is really to alert people that there may be something, and therefore the very high sensitivity performance of this sort of test will really help in making it accessible and meaningfully accessible to those. So terrific. I'm going to stop there on my specific questions, and thank you once again for giving us, first of all, the terrific work that you're doing, and I'm sure there'll be other questions when we come back to engage with our summit participants. I'm going to now move to our second panelist, Ryo Sugimura, who's going to discuss his project on reverse inflammaging by target nuclear PD-L1 in inflammatory macrophages. Ryo, over to you, and thank you for sharing your work with us, and congratulations on your award.
Yeah, thank you very much. So this is Ryo Sugimura from HKU, Hong Kong University. I would like to present my study, more like a future goal, including because I'm a PI for over 3 years now. So kind of presenting our entire view and what we are doing for longevity. So I was originally hematologist, medical background, studying hematology and stem cells over 20 years now. And yes, working on immunology for last like 5 to 6 years. So my background in stem cells, hematology, and immunology, seeing this longevity, especially aging, is an issue of systemic dysregulation of immune reaction, heterologous reaction. So what I mean by heterologous is, if you see those inflammation aging, it's kind of overactivation of inflammation in certain part of the body, but actually you are not having enough immune reaction in other parts of the body. So this is imbalance of your resource for immune reaction, inflammation. So this is kind of I view aging and a potentially important causation of why our systemic level of organ dysregulation happens is such imbalance of resource for immune reaction. So saying that, my laboratory working on bioengineer, and tackle with taking the balance back of immune system. So how to do this? So as a stem cell biologist, working on hematopoietic stem cells, human iPSCs, we can engineer and generate various types of immunocell. And when I opened up my laboratory in HKU, Hong Kong, I paid attention to macrophage, a type of innate immunocell. Why? Because macrophage is very abundant in our tissue. And this is a front line of our encounter with pathogen or like cancers. So basically macrophage touch, sense, reacts, and order your T cell, then eventually B cells and all other type of immunocell. So if you be able to regulate what macrophage does, it will be a very important step to gain back the balance of your immune system. So I have been starting from cancer model. I see this is one of very interesting immunodysregulation because in cancer, especially in solid tumor, immunologically very cold, where supposed to happen T cell reaction, somehow cancer, they suppress. So why they do? It's basically tumor microenvironment. And one of the most notorious factor is TGF beta, which has been produced by cancer itself, as well as surrounding fibroblast. And this is a culprit of down-regulating T cell reaction and also silencing your macrophage front line. So we are a team of immunocell engineer, as well as tumor microenvironment analysis in single-cell seq, a lot of special technology and single-cell sequencing technology we employ. And how we are trying to overcome this barrier of immunosuppressor, TGF beta, is we let macrophage, one, specifically target cancer. Yes, it's done by T cell people, CAR-T, chimeric antigen receptor, and we can educate macrophage, go to cancer, interact with cancer cell. But this is not enough because in tumor, that environment, you have so much of TGF, you have to block. So another trick we do is let those macrophage secrete fragments of antibody which block, neutralize TGF beta. So this is what we call antibody secreting CAR macrophage. We are working on like hepatocellular carcinoma model in animals, and we also invented new type of organoid. That project I proposed for this longevity award is actually one of our key observations in tumor microenvironment, where macrophage would be using signal of PD-L1 and facilitate its inflammatory reaction. But this is basically a mechanism. So my major aspect of laboratory is taking engineer approach and tackle its mechanism and how to revert back, say, inflammatory reaction in the tumor. So now I talk about cancer, but we recently put on the second step, autoimmune disease. So this is another spectrum, or opposite end of the spectrum. In example, MS, multiple sclerosis. It's kind of autoimmune disease affect your spine and brain. And opposite to cancer, there you are lacking a mechanism to block immune system, namely TGF beta, which is high in your physiological spine or brain. But somehow in this disease, MS, you wouldn't have so much expression. Actually, they are actively suppressed. So we can take very similar approach to an opposite end of application that you let your macrophage target surrounding autoimmune B cell, because one of the causative, culprit of autoimmune disease, they are producing autoantibody. But they are now missing immunosuppressor, namely TGF-beta. So can you supply? It's the simple way is, can you let them circulate? So this is ongoing and very interesting venue of using macrophage as a front line of the reaction. And now we take macrophage as front line of engineer and change balance of immune system. So third part, final part is, so now I talk about application in both opposite ends of immunological dysregulation. Then can we democratize? Basically make this application much easy going for anyone, because we do know CAR-T or any form of stem cell therapy. It's so expensive, may not be so much applicable for many people. So how to tackle this? We actually have a very good example in SARS-CoV-2, that messenger RNA vaccine, that you have genetic elements delivered to lymph nodes, specifically ignites kind of immunogenicity against SARS-CoV-2 spike protein. You basically educate your immune system in systemic level. So my new approach actually developing in my laboratory is, can we take those cell therapy for our engineer effort we have done ex vivo to in vivo systemic or target delivery of genetic elements, as we have done for SARS-CoV-2 messenger RNA vaccine. But if you can apply same idea of, you kind of program where those lipid nanoparticles or vehicles go to specific tissue, and what kind of elements you provide, either CAR, antibody fragments, or cytokine, you will be able to program your immune system in each tissue of target. And this will be a way to bring back the balance of your immune system in more feasible way for other people, a majority of people. So that's basically my part. Thank you very much. I'm happy to take any question.
Thanks so much, Rio. And what a fascinating area that you're dealing with in what I would humbly call sort of the young and emerging science related to immunology. And as everyone on the call will remember and know personally, the challenges of regulating the immune system with respect to SARS-CoV-2, either arising from infection or vaccination, were absolutely fascinating areas of frontier science during the pandemic. And I think where your research is headed is really to very, very exciting frontiers of precision immunology, as you say, really rebalancing immunologic dysregulation, and potentially doing that with specificity to different types of tissues, but also perhaps specificity to different types of immune systems, recognizing that not everybody has exactly the same immune functions at each stage of life as well. So very, very interesting. I have no specific questions on that. I'm not an immunologist per se, but I'm sure there will be, particularly with respect to the translational dimensions of what you're considering. And when I say translational, you've got some terrific, what I would call frontier biologic mechanistic opportunities that you're developing, getting those into the population at large and accessible, I'm sure are challenges that you've thought about and others may have questions on. So I think what we'll do, Rio and Jakkrapatr, first, thank you for your presentations, absolutely excellent. And let me say on behalf of the Catalyst Award panel that I was co-chairing with my co-chair, Dr. Roderick Pettigrew, this gives you a taste of what I think were a broader group of absolutely brilliant ideas that are emerging. We had a couple of other panelists for this session who are unfortunately unable to join, but you would have seen in their talks, one focusing on the health workforce in the aging population, and another working on issues related to dementia. That there is just a wealth of emerging science that is part of the excitement associated with this award. So with that, I'm going to ask a couple of questions, and I will of our panelists, and then if people from the audience who are listening have questions, please use the chat, and those will be directed to me. And so I'm happy to introduce those questions to our panelists as they emerge. But let me begin with a general question to both of you. And this is really a little bit more on personalizing your stories. As you move forward in science, great ideas always hit roadblocks. I'm sure you've been disappointed. You found out something that you were expecting didn't work. So it'd be great if you could each talk about one or two major challenges that you faced. Something that wasn't expected, something that really had you perplexed, and how you manage that. And it doesn't necessarily have to be something you solved. It could be something that you're still trying to figure out. But it'd be great, I think, for those who are listening to listen to what were some of those big roadblocks or hurdles that you had to get over, and it wasn't necessarily obvious, or it wasn't part of your initial plan or how you thought the science would roll out. So Jakkrapatr, over to you.
Yeah, I would like to start by mention first of all, the most important thing is the yield rate of my lab ownership, and actually it's both sides, from lab ownership and from the reader circuit. I mean, what I expect, as we know that making one chip or one PCB board or for this reader circuit is not an easy process, and the yield rate will it's the main thing that we have to consider, because if the yield rate is low, it means that of course we cannot commercialize or cannot make our product in mass production. And that is the main problem that we face, because in order to control the quality of each lab ownership is the hardest thing that we throughout the last year we trying to solve all the problem, and we trying to tackle all of this problem. And yeah, of course, thanks to the Catalyst Award that give us the funding that, and this one is pretty important fund, is because it make us like a really large step toward the final result what we expect in the future. And yeah, that is all what I want to talk about for my challenge.
Thank you, Jakkrapatr. Rio, what were some of your one or two of your major hurdles or challenges that you've encountered?
Mhm. So let me start from a technical challenge that we faced last 3 years since beginning of my laboratory. So I mean, when I set up my laboratory, it's kind of very easy thought that, okay, let's say we can do TAR macrophage from iPSC. And yes, I have 10 years of experience in iPSC hematopoietic differentiation. We do the macrophage is one of easiest cell type. And chimeric antigen receptor CAR has been already used so much since late 1980 to T cell. Even clinical trial, so much well done. CAR macrophage new, but it's well established in primary macrophage. So that was a very initial setting, and I kind of thought, oh, that easy going, low hanging tree. Reality was not. So one obstacle, I mean technical point, because in case our audience has anyone interested in cell therapy, iPSC engineer, macrophage engineer, will be good lesson. I mean, one, iPSC macrophage, they didn't write like very established chimeric antigen receptor perfectly used for T cell, but not in our macrophage. They kind of like put back those macrophage, retract from surface, and stay in Golgi body like a garbage. So that's one major issue we solved. One. Then two, yes, we are actually kind of improving that aspect. We can make iPSC CAR macrophage. But those macrophage still cell number, as a manufacture issue for cell therapy, you need millions or billions of cells for patients. Just doing this for iPSC was so tough for technical rights because their initial cell number and differentiation efficiency and those monocytes, macrophage don't divide much on Petri dish. So that was a second and biggest hurdle we kind of encountered last year. But we are so lucky. We had a postdoc from neuroscience, completely different field. And he has a very interesting technology. Using AAV, adenovirus associated vehicle, Cas9 deliver, specifically insert very complex structure in like any desired loci. He proposed, hey, Rio, why don't you do cold blood, real hematopoietic stem cell? Yes, we know perfectly cold blood HSPC is fine. They divide like crazy. Super duper easy to make macrophage larger cell number than that you do with macrophage. But as stem cell guy, I have been also using HSPC, mouse bone marrow, cold blood last 10 years. We don't know how difficult it is to gene edit. Put precise CAR in precise loci. What sounds nightmare. iPSC was way easier. So I was initially kind of, hey, what the things are saying. But this neuroscience postdoc convinced me, let's try AAV, Cas9, electroporation, and see efficiency. And it was over 70%. Yes, after a couple of trials. Optimization using DNA PK inhibitor, that's also known in his field. And he brought those kind of recipe to our field. It dramatically improved our generation. And now we are proposing primary cord blood hematopoietic stem cell as quite superior source of engineered cell type, immunocell engineering. So that's basically two technical barriers we encountered before, and we are basically addressing as an entire team effort of laboratory. And I do appreciate we have staff from different field, different background from PI. Yeah, thank you very much.
Yeah, thank you, Rio, for that. And I just want to acknowledge this sort of I won't call it serendipitous, but really you found a solution to your problem from an unlikely place and an unlikely discipline. It was a neuroscientist working with an AAV, as you say, and who actually, addressed your cell production problem in a way that probably wouldn't have come from you guys alone. And so I just want to stress this the opportunity of convergent disciplines that sometimes bring and help make those synaptic connections to get over hurdles as part of what I think is really creative in this whole process of the awards. I have another question that I'd like to both of you, and Jakkrapatr, you mentioned the financing. I'm an economist in addition to a clinician for all my sins. So when you mentioned the award value of $50,000, you said that this is going to be very helpful. Now, getting products to market it takes a long time, but also takes a lot of money. And often the institutions that do that are ones beyond your laboratories where you've been doing this work. So it would be great for me to hear two things. First, have you thought about plans on getting your products from the lab to the market? Have you had some thoughts? And two, can you reflect on your own training? And in any of your training, were you given an opportunity to understand what's involved in getting great discoveries in the lab to the market? And please be very honest. This is not to try and assess your institutions, whether they've done a good job or not. I'm just curious for you to reflect on the extent to which your own training, you've had fantastic training in your respective disciplines, but did you ever have the opportunity in that training to begin to understand what's involved in taking brilliant discoveries like yours from the lab to the market? So those are two questions, and we'll reverse it here. Well, Jakkrapatr will give you a little time to think on that. Rio, over to you. Rio, I'm going to ask you to go.
Yeah, sure. Yeah, I have already answered. Yeah, because this is exactly we are working on for commercialization of our chimeric antigen receptor macrophage from stem cell source to the market. And yes, also Hong Kong is very like Singapore. We are very much focused on like bench to commercial. And we already working on making spin-off company. And we do have one help, kind of Hong Kong resource money to especially support this type of work, commercialization or making spin-off company. But so that's answer one. Then answer two, regarding my background training. Yes, I am also clinician in training, MD. And then my PhD in hematology stem cell. So I have to confess, nothing, zero training. Even my postdoc in Harvard, I was a stem cell guy, iPSC, medium change, gene transduction, inject into mice, flow cytometry, single cell RNA-seq. Nothing about marketing. So yes, in Hong Kong, we are very much encouraged to do marketing. But PI has zero idea, especially at least me, I have zero idea. So very interesting moment came last year. So I kind of pitched and I got a very small fund to support my activity idea of commercialization, spin-off. But yes, I have some like a small money, zero strategy. And I was basically like sitting down on my money. What to maybe just buy laboratory reagents, and that's it. But then exchange student from Germany, Heidelberg, came to my laboratory. He is MD background, medical student, amazing laboratory person and conducting bunch of experiments. One day, we invited a kind of business background person like Anthony, and he was educating us how to think about making money. And then suddenly, this student asking very sharp, make sense questions, like inside economy person. And I was very surprised and asked him, what is your identity? And actually turns out this medical student originally from economy background, I mean, Department of Economy graduate. He really wants to do startup biotech. And before that, he wants just an MD degree and do some training of science and then move on to industry. So that's his plan. So I'm very, again, lucky to have someone from completely different background. And this person kind of educated me and my entire team and connect to right people, I mean, departments for these business type activity. To me, still myself have zero idea, I mean, zero knowledge. So I very much have to know who to connect and what is structure I get a support. And this person basically led me to this area. Yeah, thank you very much.
Great, thanks, Rio. And just again, the international connections there, I think, are really interesting, serendipitous in some level, but also the interdisciplinary. Jakkrapatr, over to you on those two fronts.
Yeah, in order to answer the first question is, of course, every step. I mean, since the first day that I try to enable this project, and try to commercialize this project. One thing that I always keep in mind is of course, if you see the paper in the field of electrochemical sensor or biosensor, you found that we have several research group that already finished or already complete the really high sensitivities cardiac biomarker detection, but none of them can really commercialize it, and that is because of many problems like the shelf life, the cost of the manufacturing process, and moreover, when we try to make this kind of reader circuit or chip, of course, we cannot finish the whole thing within our university. And that is why we need to ask our outsource. And sometimes our outsource cannot provide, I mean, it takes so long to complete some of the, like one of the version of the lab on the chip that we design or the leader circuit. And it might take, like for this one, it might take 4 months in order to manufacturing the prototype of the leader circuit. And that is all of them is the challenge that we face. But of course that for the commercializing this product is the one that we always keep in mind, and that is why and we know that we are technology university. That is why we try to like enable the idea and theory that we have, that researcher find and export it into the product. And I think that is the strength of our university. And for the second question, pardon, could you repeat the question of your second question again?
Just whether you had any training in commercialization as part of your formation.
For those part, like my professor, like he have some company in under his control, but to be honest, we don't have those kind of training in our school yet.
Understand entirely, and it's just to say it's something that I'm particularly interested in myself, which is trying to address that shortfall in training and create opportunities for intrepid discovery scientists like yourselves to understand that long road to getting it from the bench to the bedside, to make sure, or to patients' homes or to everyday life, so that people can benefit from the tremendously exciting and innovative work that you're doing. So we've come to the end of our time. I'd like to thank both of you for fantastically interesting presentations. Congratulations on your awards. I think you've provided a sense of the inspiration and the opportunity associated with your respective work. Wish you all good luck and ability to climb over those tough hurdles ahead. Given what you've accomplished thus far, I'm confident that you'll continue. And thank you today for sharing those with everybody who has had the opportunity to listen to you. Thank you very much. And with that, we'll conclude this panel.